Well, thanks very much for joining us on this very special and unique research round. And so this is our research conference that will feature a special speaker, Dr. Sarah Dick, today. But this is also one of our first hybrid events. And thank you so much for joining us, both in person here as well as virtually on the virtual platform. And this is because today we're going to have Dr. Dick to give us a special update in terms of the exciting research that she's doing. But this is also her visit to the Heart Institute today in person. So we're absolutely privileged to have her here. And for those in the room, the questions, following Dr. Dick's presentation can be posed directly in person using the microphone on the side there. And because this is the way we can project this onto the virtual platform. And however, for those who are joining us virtually, please use your Q&A feature on your screen to pose your questions, which we can actually entertain them following the lecture itself. And but also you can use the hands up feature in which we can invite you to actually join in on the panel discussion, and you can pose the questions directly. And if you have any technical features, use the chat feature as before. So we are absolutely delighted today that Dr. Sarah Dick is able to join us and also, discuss a lot of the research opportunities with our research colleagues here at the Heart Institute as well as the university. She currently is a postdoctoral fellow at the Toronto General Hospital Research Institute and at the Ted Rogers Center for Heart Research. She's working in the lab of Dr. Slava Eppelman, and many of you know that he is not only a good friend and colleague of investigators here at the Institute, he's one of the top immunology researchers in the world, and certainly in terms of cardiac immunology, he's really actually at the cutting edge. And Sarah holds a CIHR fellowship for her work in the field of cardiac immunology. And she completed her PhD in the field of regenerative medicine here at the Ottawa Hospital Research Institute, the Sprott Center, with Dr. Lynn McGinney. And she then spent a period of time developing caspase-3 conditional knockout mouse at the Institut Cochin in Paris, in France. I guess there's a lot of hot politics going on there in France today. And Sarah's seminal work on tissue-resident macrophages really actually constitutes the bulk of her current work in Dr. Eppelman's lab. And she has identified novel populations in the heart are critical regulators of cardiac repair. And her research focused on defining the framework of cardio-immune interplay at the systems level to decipher the roles of macrophages in the control of cardiovascular disease, such as cardiac remodeling, heart failure, and really many other conditions. Her research combines techniques of molecular and cell biology, genetic fate mapping, single-cell RNA-seq, and bioinformatics to really help to identify the populations that have important homeostatic and reparative functions and how those may actually be dysregulated in the setting of disease and in leading up to chronic inflammation. And this is, of course, not only give us the means to interrogate the system in the setting of cardiovascular disease, but in fact, in many other settings that includes abnormal system response. So it's really applicable across many, many disease processes, including peripheral muscle, for example. And Sarah publishes in some of the highest impact journals in the field and is really a beacon in terms of innovation. And we look forward to her presentation today, and that's harnessing tissue-resident macrophages to improve cardiac function following ischemic injury. Sarah, thank you so much.
For that really nice introduction and for hosting me here at the Heart Institute today. It's been a really great visit so far. And thanks to all of you tuning in on Zoom. This audience doesn't really need an introduction to the heart, but just so we're all on the same page, heart disease is the leading cause of death worldwide and the leading cause of hospitalizations in Canada. Now, according to the CDC, one person dies every 36 seconds in the United States from cardiovascular disease. Heart disease is also a strong comorbidity factor in the general population, including those with COVID-19. It's estimated that in any given year, Canadians with heart disease are up to 6 times more likely to die of any cause than those without heart disease. Of heart failure is coronary artery disease. Now, as the name suggests, this is caused by narrowing of the coronary arteries, leading to a reduction in blood supply to the heart muscle, and can often result in plaque rupture and complete occlusion of the heart tissue as a heart attack or a myocardial infarction, and results in the cell death of the cardiac tissue, resulting in inflammation and infarct expansion. This then leads to fibrosis and scar formation, and eventually to elongation and dilation of the ventricle, resulting in adaptive cardiac remodeling, such as myocyte hypertrophy. And this leads to congestive heart failure. Now, importantly, the healing from cardiac injury is modulated by innate immune cells called macrophages. These are a mononuclear phagocyte, which essentially means they are big eaters. So they are innate immune sentinels that go around engulfing and digesting dead cells and cellular debris. They also patrol tissues for pathogens and present foreign antigens to T cells to initiate an inflammatory immune response. Homeostasis. So specifically in the heart, they've been shown to play a role in angiogenesis and coronary development. They've been shown to facilitate electrical conduction through the formation of gap junctions. And more recently, they play a role in metabolism through eating the mitochondrial from the cardiomyocytes themselves. Reparative responses, such as cardiac regeneration in the neonate. They're anti-inflammatory mediators. And as I'm going to show you today, they have important roles in adaptive tissue remodeling. Have been found in every tissue of the body, where they receive tissue-specific signals to produce these specialized homeostatic functions. So in addition to their general roles of inflammation and wound repair. Now, historically, we've been taught that macrophages come from hematopoietic stem cells from the bone marrow through a process of definitive hematopoiesis. And upon entering the tissue, they differentiate into this wide array of tissue macrophages. But what we now know is that the majority of these tissue macrophages come from embryonic sources, such as the yolk sac and fetal liver, through process of primitive hematopoiesis. In both animal models and clinical data, that excessive monocyte and macrophage expansion impaired tissue repair. Suppress monocyte and macrophage expansion or their non-specific depletion also limited tissue repair, thus creating a paradox in the field based on the assumption that all macrophages are created equally, which leads to the hypothesis that different types of macrophages exist within the cardiac tissue. Dr. Eppelman published his work highlighting a level of heterogeneity in cardiac macrophages pertaining to ontogeny. So during development, yolk sac macrophages seed the tissue and take up an MHC2 low phenotype. These are joined a little later on by fetal monocyte-derived macrophages, and a proportion of these will upregulate MHC2 and become an MHC2 high macrophage. And these macrophages have functions in terms of phagocytosis, angiogenesis, and regeneration. Now, a little later on, after birth and in the adults, we have the contribution of bone marrow-derived adult monocytes, which enter the tissue and either downregulate CCR2 to take up a phenotype very similar to the MHC2 low and MHC2 high macrophages, or they maintain CCR2 expression and become a population of CCR2 positive macrophages. And these CCR2 positive macrophages express genes important for inflammation. By a lot of interest into these different macrophage populations and how they differentially contribute to inflammation and repair. So my aim was to characterize the cardiac macrophage subsets and their roles in heart injury and repair. Data today that I did in the setting of MI that shows tissue-resident macrophages self-proliferate and are important for the repair process, whereas the monocyte-derived macrophages are inflammatory and inhibit this reparative process. Understanding of the transcriptional differences among the macrophage subsets in the heart, I performed single-cell RNA sequencing on sorted CD64 and CD11b positive macrophages, and we used the 10x genomic platform, and Sarani Jat helped me with some bioinformatic work on this project. Of 1,200 single cells, we see they form these 5 clusters as visualized here on the T-SNE plot, where each dot represents a cell. And these represent 5 transcriptionally distinct subsets. And we can visualize that here on a heat map. Now I've pulled out a number of genes here on the side that we can then use to annotate our clusters. So this first cluster is a small cluster of monocytes present that express the monocyte gene ACE and lack mature macrophage markers such as C1qa and FCGR1. This other cluster here in blue is a small cluster of ISG macrophages, named so because they express a high number of interferon-stimulated genes such as IFIT3, RF7, and IFIT1. Uniquely express the markers TIMD4, LIVE1, and folate receptor 2, as well as the growth factor IGF-1. So I'm going to refer to these as TIMD4 positive macrophages. Lack these markers and expressed high levels of the CCR2 gene, and so we're naming these the CCR2 positive macrophages. And this cluster in the middle lacked these markers and expressed high levels of the antigen processing gene MHC2, and we will be referring to these as MHC2 high macrophages. Complemental relationship exists between the characterized macrophage subsets. We performed expression-based ordering of differentially expressed genes among the T-SNE clusters using a software package called Monocle, which orders these cells along a pseudo time. So you can see how these genes change over the pseudo time within our different cell populations. We can then pull out a few of these genes. Here we've pseudo-colored the cells with the color from their cluster with the gene expression on the y-axis and pseudotime along the x-axis. And you can see this increased expression of TIMD4 and LIVE1 with this concurrent downward expression of CCR2. Different package called empath, which also allows us to infer developmental relationships in a more specific way with the number on the arrows indicating the strength of this relationship. And this allowed us to infer a linear relationship between monocytes, the CCR2 macrophages, the MHC2 high macrophages, and the TIMD4 macrophages. And interestingly, the ISG macrophages seem to have a relationship directly with the CCR2 macrophages, as opposed to becoming directly off the monocytes. To these subsets have specific functions, we performed pathway analysis on the differentially expressed genes from each subset. And we found these TIMD4 macrophages had pathways involving receptor-mediated endocytosis, regulation of vascular development and wound healing, as well as fibroblast proliferation and response to muscle stretch. Macrophages had pathways involved in antigen processing and presentation, as well as positive regulation of T-cell activation. And the CCR2 macrophages, as we predicted to have a more inflammatory role, does appear to have a positive regulation with inflammatory response and defense response. These are novel single-cell RNA-seq cell surface markers to track these populations using flow cytometry in the animal. So here we can identify our two CCR2 negative populations, our TIMD4 positive and our MHC2 high, which are TIMD4 negative, and our one CCR2 positive population. And if we use these to go back and look at how they respond to the original subsets identified by Dr. Eppelman, we see that the MHC2 highs, of course, are almost entirely MHC2 high. And these TIMD4s actually are mainly MHC2 low, although some do express some MHC2 high expression, which we do see reflected in the single cell data a little bit. Populations over the life of the animal, we see that they start out with the subset, the TIMD4 subset majority at E14.5, and they maintain this majority over the life of the animal. And if these populations are resident versus recruited from circulating monocytes, we developed a fate mapping mouse model using the CX3CR1 CRE-ER mouse crossed to a ROSA TD reporter. So following tamoxifen or TAM administration, there's a permanent recombination event leading to TD expression in all CX3CR1 expressing monocytes and macrophages. And so we did a pulse chase where we can pulse them and highly label all these cells and then chase them for up to 6 weeks. And what we see is that the monocytes lose this label. Since monocyte progenitors don't express CX3CR1, any monocyte replacing those will be TD negative. Now, interestingly, the CCR2 positive macrophages also lost the TD signal over the 6-week time point. However, the CCR2 negative cardiac macrophages maintain this signal for a very long time, as long as we've looked in a 2-year mouse. And so we can use the system to specifically label the resident cardiac macrophages that are maintained via self-renewal. And so next I examined how these macrophages changed in the setting of cardiac injury with specific attention to these resident populations, with the hypothesis that they play an important role in tissue healing. Performed a surgical model of myocardial infarction or heart attack by permanent ligation of the left anterior descending or LAD artery. We performed flow cytometry on the ischemic zone, which included both the infarct and the peri-infarct region. And this work was done in collaboration with master student Jillian. A massive recruitment of TD negative macrophages in the infarct zone, as measured by TD negative macrophages per milligram of tissue. What was really interesting is that in the setting of injury, we actually see a loss in both the percent of TD positives, as well as the absolute number of macrophages per milligram of tissue of the TD positives early on at day 2. Now, these slowly come back to numbers closely resembling control, but they remain very much a minority population, remaining 22% of the population at day 28. To the neonate. So neonatal mice that are less than P7 have the ability to regenerate their hearts post-injury. However, this ability is lost when macrophages are non-specifically depleted, indicating that macrophages play a key role in this regenerative response. Now, in a diphtheria toxin receptor model of cardiac injury, Dr. Levine has shown that the neonatal mice have a majority of CCR2 negative MHC2 low macrophages. However, in the setting of injury, it's this population that expands in number. And as mentioned, this is in contrast to the adult. Influx of CCR2 positive macrophages. And so this is important in the different way that the neonate and the adult respond to injury. Interested in understanding how the CCR2 negative macrophages are regulated and expanding in the infarcted tissue. So first, to confirm our flow cytometric findings, as well as to gain a better spatial resolution of the infarct and peri-infarct regions, we performed immunofluorescence imaging. So here, all macrophages are labeled green for CD68, with the resident macrophages co-expressing the TD reporter. And we're able to confirm that early loss of resident macrophages at day 2 in the infarct region. But what's really interesting to us was this local expansion in the peri-infarct region of the resident macrophages. Yeah, which subset of tissue resident macrophages were expanding? We included a live 1 stain to mark the TIMD4 positive population, along with the TD reporter. And so, although we saw no difference actually between the live 1 positive in red or in green and the live 1 negative in red, and this is in the infarct region, in the peri-infarct region, we saw that it's actually the live 1 negatives, or the TIMD4 negatives, that are preferentially expanding in the peri-infarct region, suggesting they might have a functional role in this area. Mechanism of expansion is proliferation. So to assess proliferation in the infarct tissue, we labeled mice with a BRDU pulse 2 hours prior to isolation. Sent BRDU positive cells by flow cytometry, we see there is this early proliferative burst post-injury in the resident macrophages at day 2 and day 4 post-MI. This is pretty similar between the TIMD4 positive and the MHC2 high macrophages, with a slight advantage to the MHC2 high macrophages, suggesting that they might be proliferating in the peri-infarct zone, as we saw by immunofluorescence. Macrophages, we see that they do also have a proliferative burst at day 4, a little slightly delayed compared to the resident. But interestingly, it looks like the resident and recruited macrophages use a common mechanism to control cell numbers through proliferation. Emphasize that it was competition with recruited macrophages was controlling the resident macrophage numbers in the injured heart. So to test this, we looked at the resident macrophage re-expansion in the setting of the monocyte deficient CCR2 knockout mouse. And you can see here by flow cytometry, they just have a lot less CD64 positive macrophages. The monocyte recruitment, we see actually an accelerated resident macrophage proliferative re-expansion, with 10% of macrophages proliferating in the knockout mouse at day 4 post-MI. An enhanced numerical recovery of the resident macrophages post-injury, and actually just more macrophages in general. Recruited macrophages limits the ability of the resident macrophages to expand in the infarct zone. And understanding how these recruited macrophages regulate the resident macrophage numbers will be critical to promoting their presence in the injured heart. And this is an area I'm very interested in following up on. Understanding of the recruited macrophage heterogeneity in the infarcted heart, we did single-cell RNA-seq on sorted cardiac macrophages isolated 11 days post-MI. And this is compared to their littermate controls. So we included almost 2,000 control cells and over 4,500 post-MI cells. And as you can see, a number of these post-MI cells fall within a similar gait as the steady-state macrophages that I've previously described to you. However, the majority of these cells are actually MI-specific macrophage clusters. Just cluster 6 here, which includes monocytes differentiating into macrophages. And these differentiated macrophages have a high HIF1 alpha and other hypoxia response gene signature to them. Now, I'm not going to go into too much detail today on the recruited macrophage populations because we honestly don't know too much about them. However, we're very interested in understanding these cells, what they're doing, how this heterogeneity changes over the course of injury repair, with the ultimate goal of identifying how these changes may predict outcome or the course of disease. It's back on the resident populations and work I've done to understand the specific roles that these cells play in altering cardiac repair. So to test this, I took advantage of the genetic fate mapping mouse, the CX3CR1 CRE-ER with the TD reporter, this time with the diphtheria toxin receptor in one of the ROSA alleles, which will allow for the selective depletion following our pulse chase strategy of just the resident population. And as you can see, this results in good knockdown or depletion, I should say, of the resident macrophages, however, not the CCR2 population, not the lymphocytes, neutrophils, or monocytes, and importantly, not the monocyte-derived macrophages. Experiment where we gave diphtheria toxin daily, starting 1 day prior to MI, and for 35 days. We confirmed that we maintained depression of the resident macrophage numbers. Left echocardiogram to measure ejection fraction as well as histology. The hearts depleted of the resident macrophages only is a decrease in left ventricular ejection fraction when compared between day 7 and day 28. We did not see this with the MI control. Tissue samples isolated to day 35, we looked at WGA staining as a measure to measure hypertrophy, which essentially is the thickening or enlargement of the muscle tissue resulting from an increase in cardiomyocyte cell size. So measuring cardiomyocyte cell size here, we saw that following MI, we do see an increase in cell size or cardiac hypertrophy, and this was increased in the hearts depleted of resident macrophages. Fibrosis using a serous red fast green stain, and again saw in macrophage depleted hearts an increase in fibrosis in the peri-infarct region. Now, importantly, both cardiac hypertrophy and fibrosis are measures of adverse cardiac remodeling, suggesting two mechanisms by which the resident macrophages are altering cardiac function. What I've showed you in the first half here on the heart, we have these different macrophage subtypes that have their own inherent functions and roles in inflammation and tissue repair. The CCR2 negative macrophages are resident and maintained by self-renewal. They express growth factors important for homeostasis and repair, such as IGF-1. And the resident macrophages are lost post-MI in the adult, where they have important roles in tissue healing. And as I showed you in some preliminary data, their re-expansion is limited by monocyte recruitment. On the other hand, the CCR2 positive macrophages are continually replaced by circulating monocytes and express inflammatory genes. Differentiating between the macrophage subtypes is important. There we go. Key to diagnosis and treatment of pathologies caused by their dysregulation, such as chronic inflammatory diseases and in aging, so that we can target the right subset. For example, broadly targeting immune cells in chronic heart failure has led to mixed and sometimes detrimental results. So ideally, we want to eliminate the inflammatory macrophages while promoting the presence of the reparative ones. This is true for all organs, not just the heart. So in the second half of my talk here, I'm going to share some really exciting findings I've been working on to examine if a common macrophage framework exists across organs. And this was a really collaborative effort that started between myself and postdoc in Dr. Tak-Mac's lab, Rob there, while we were both trying to optimize our single cell techniques for our tissue of interest. So the idea we came up with was, does a common framework exist, and can we make a comprehensive map of tissue resident macrophage heterogeneity across several tissues? And this is based on what we were seeing with our flow cytometry experiments at the time. So since then, this idea has grown. We've recruited the help of graduate students Anthony and Hamira with additional bioinformatics support by Sarah. And so this really addresses one of the challenges in the myeloid field, which is that although several macrophage subpopulations are identified, within individual organs, there lacks an ability of findings from one study to inform another. And this is due to different approaches and nomenclatures used. So for example, is macrophages from one tissue similar in some way to macrophages from another tissue? And this is true across species as well. So are macrophages, how similar or how different are macrophages in the mouse to the human to help facilitate that movement from bench to bedside? And so our question is there an underlying organization that can inform a common starting point for studying tissue macrophage heterogeneity in biology? So here we focused on commonalities between the macrophage subsets in both mice and humans. And we performed single cell RNA-seq to reveal 3 macrophage subpopulations with a small core gene signature across organs. Organization tracks with biological properties such as monocyte replacement. Data on how we arrived at our core gene signature, what we did was to sort CD64, CD11b positive macrophages from 5 mouse tissues and perform single cell RNA-seq, again using the 10x genomics platform. And then following filtering and quality control, we were able to visualize this using dimensionality reduction using UMAP. I've previously shown you, we have 3 clusters representing 3 macrophage subsets with reciprocal expression of TIMD4 and CCR2. Now, similarly, when we zoomed into the liver, we again see 3 subsets here, with the large proportion of them being the well-known liver Kupffer cells. Again, in the kidney, we see 3 subsets with a smaller TIMD4 population and the majority of them being the CCR2 positives. We have a little bit of a different situation where we have a large proportion of the well-known alveolar macrophages, and these are different than the interstitial macrophages in the lung that have been well characterized. However, if we zoom in on the interstitial macrophages, we again see our 3 subsets with reciprocal expression of TIMD4 and CCR2. The majority of macrophages in the brain are microglia, but there's a newly identified population that are more perivascular called border-associated macrophages, or BAMs. And these again can be subsetted into our 3 populations, with TIMD4 and CCR2 expression again being found. Looks like differential expression of TIMD4 and CCR2 tracks with our 3 macrophage subpopulations across tissues. But importantly, to what extent are macrophage subsets similar to each other across tissues? Is just the few markers or general globalization? So we took differentially expressed genes from each organ and looked for genes shared across the similar clusters. And what we found was that although the majority of these genes were tissue specific, so meaning present in only 1 organ, up to 10% of these were shared amongst the 3 organs. And this allowed us to come to a small shared gene profile to define these 3 macrophage subsets across organs. So here I'm going to sort of switch a little bit to calling this TIMD4 positive subset TLF positive, just to be more inclusive, that it's really the TIMD4 or LIVE1 and/or folate receptor 2 that defines this population across tissues and species. Random forest modeling, which is a machine learning technique to predict which cluster a cell will fall into based on its expression of 4 cell surface markers, TIMD4, LIVE1, folate receptor 2, and CCR2. And what we found was that these 4 cell surface markers can predict this with actually very high accuracy. Exists among similar macrophage subsets. We again performed pathway analysis and found 2 common pathways for the TLF positive macrophages, including vesicle-mediated transport and receptor-mediated endocytosis. We found 3 common pathways for the CCR2, including immune effector process, leukocyte degranulation, and myeloid leukocyte activation. With the MHC2 high macrophages having a more tissue-specific flavor to them, I would say. There wasn't a single pathway that was exclusive across all 5 organs, but you can see trends towards a more, cell migration or regulation of cellular component movements Populations are enriched in a core set of pathways across organs, and this sort of suggests functional similarities. Heterogeneity also exists within human tissues. We obtain heart tissues through collaborations at Sick Kids, the heart, and we downloaded publicly available single-cell RNA-seq data from the other organs. Now, I know there's a lot of data on this slide, but I really want to draw your attention to the fact that there's, again, 3 macrophage subsets identified in each of these tissues. And this was really exciting for us, as we also found our cell surface markers, Live1, TIMD4, and folate receptor, was also conserved in the human tissues, with one of the populations also expressing CCR2. So it appears that the subset defining genes that we found in the mouse are consistent in the human as well. Differentially expressed genes from the subsets between mouse and humans, we see that across tissues, the highest level of transcriptional similarity is among that TLF positive subset. And so now when gaining tissue macrophages across the different tissues, with specific focus here on the CD11b high interstitial macrophages in the lung and the CD45 high BAMs in the brain, we can stratify these macrophages into 3 macrophage populations by flow cytometry. And we can track how these populations change over time, with a number of them starting out with a TLF positive subset majority. However, this composition changes over the life of the animal, with a noticeable loss of the TLF subset in the kidney, which reflects our single-cell RNA-seq data. Holds true in humans, so we were able to obtain fresh heart and lung tissues thanks to our surgical collaborators. And again, we could identify all 3 macrophage subsets by flow cytometry in human tissues as well. Identification of these 3 transcriptionally similar subsets, we asked, does this framework track with common biological properties? So here we focus our attention on 2 aspects of macrophage homeostasis: ontogeny and turnover. Ontogeny, we look directly at macrophages in the yolk sac of E14.5 embryos. And interestingly, we see pretty much all of the macrophages present in the yolk sac express TIMD4, Live1, and folate receptor 2, and are CCR2 negative. What was really more amazing is when we looked at single cell data from human yolk sac, we see that almost all human yolk sac macrophages also express TIMD4, Live1, and folate receptor 2, suggesting a developmental origin of these TLF positive macrophages. However, to test this experimentally, we used our genetic fate mapping mouse, again, the CX3CR1 Cree-ER with the Rosatides reporter. And this time we injected tamoxifen to pregnant females at E8.5 to specifically only label yolk sac macrophages. Now, this is an example from the heart. We see we're labeling only macrophages. And in fact, the majority of these macrophages, when we look at their subset, are TIMD4 positive. However, we are labeling a number of the MHC2 high macrophages, and this is consistent across tissues, suggesting that both these populations have a yolk sac origin. Embryonic hematopoiesis, which plays a major role a little later on in development, is the fetal liver. So analysis of the human fetal liver, single-cell RNA-seq data revealed 2 important observations to us. The first is that macrophages within the human fetal liver also express Live1, TIMD4, and folate receptor 2, similar to what we see in the adult tissue. And second, these TLF positive macrophages occupy 2 different clusters, cluster 1 here in pink and cluster 2 here in teal. Now, using a Monocle 3 program, which separates clusters based on superclusters, which are predicted to have their own single cell trajectories, we can see they actually occupy 2 different partitions here. And so if we look at the macrophages here from the monocyte-derived partition that's clustering with the monocytes, it's lagging here, what we found was that the monocytes coat upregulate macrophage genes, including live-1, TIMD4, and folate receptor 2, while downregulating monocyte genes such as FCN1 and S100A. So this suggests a dual origin for these TLF positive macrophages, with one of them being from the yolk sac and this other one being from fetal liver monocytes. Again, to experimentally test this time we used a CCR2 Cre-ER to specifically label fetal monocytes. And we injected tamoxifen at E14.5, a time when fetal liver hematopoiesis is occurring. E19.5, just before birth, we didn't see too much labeled. However, we fostered a number of these litter mates and followed them out to 3 weeks of age. And we see that the TD positive cells expand in number. And this expansion is largely due to the expansion of the TLF positive subsets derived from these fetal liver monocytes. When we inject these pups at P7, which is a time when bone marrow hematopoiesis is occurring, we see, although we have decent labeling, these macrophages don't really stick around in the tissue and are probably giving rise to the patrolling CCR2 positive monocytes and macrophages. Tracks with monocyte turnover, we performed parabiosis experiments where mice are surgically joined to share a circulatory system. And if we look at the lung as an example here, we see a number of the lung interstitial macrophages express CD45.1, suggesting they came from the donor. And alveolar macrophages, which are known to be really stable without donor input, remain 1%. If we take the interstitial macrophages and parse them into their subsets, we see that the CCR2 positive macrophages are almost entirely donor derived, whereas the TLF positives are almost entirely host derived, meaning very little input from circulating monocytes. And the MHC2 population had some about 20 to 30% of monocyte replacement. And so when we applied this technique across all tissues, again, we can observe consistently that the TLF positive macrophages lack monocyte input. The CCR2s are the highest with monocyte input, with the MHC2s being intermediate. And this is true even when we looked long-term, here at 2 consecutive time points, so 6 and 10 months. And so interestingly, what we see is that, again, we don't have very much input into the into the TIMD4 positive or the MHC2. But when we looked at 6 months to 10 months, we actually didn't see this increase at all. This little bit that did go in, we didn't see it increase. So this suggests that monocyte replacement actually reaches a plateau of maximum contribution. And that was really surprising to us. Of course, the liver is the exception here. To see how this framework is held up beyond these 5 tissues. So in addition to the heart, liver, lung, kidney, and brain I've been talking about, we assessed 12 additional tissues by flow cytometry. And this included 4 muscle tissues, 4 digestive tract tissues, 3 fat tissues, and the skin. So first we gated here on total CD64 positive macrophages, and then subsequently gating on TIMD4 and CCR2 to identify 3 similar populations in each tissue, which a sizable TIMD4 population at most. And incredibly consistently across all 17 tissues, we see that the TLF positive macrophages have the highest self-renewal capacity. So again, no maintained independent monocyte replacement. The MHC2 highs had intermediate replacement, and the CCR2 positives had high replacement. And I'm showing you parabiosis data here, but we also confirm this with genetic fate mapping. Complex heterogeneity that's emerging in the tissue macrophage field, we can organize these across tissues into a common framework of 3 subsets. Shown you today is work that I've completed in my postdoc using single-cell RNA-seq and genetic fate mapping to show that macrophage phenotype and function is dictated by ontogeny, where they come from, their subset, their environment, so steady state versus injury. Their tissue niche has a big role in their phenotype, as well as other macrophages. So I showed you some data that resident macrophage expansion post-MI is limited by the monocyte recruitment. And again, this is important because we need to accurately differentiate between the different macrophage subsets so their precise role in homeostasis, injury, and repair can be determined. And so future work in my lab will use contemporary immunologic techniques to determine the factors involved in resident macrophage self-renewal and repopulation after injury, so that we might exploit these to expand cells in the setting of injury as novel therapies to enhance repair. I'm also interested in understanding the specific roles of these resident macrophages have in skeletal muscle regeneration. And by understanding the subsets present in steady state and injury at the single cell resolution, we can begin to predict and define the growth factors in cell-cell communication that's important to this process. Knowledge gained from my previous experiments in the setting of MI to identify resident macrophage-derived factors that may influence recruited macrophage polarization upon entry into the tissues, which I'll confirm in vivo. And together with the identified environmental factors, we can then use this knowledge to promote the polarization of the recruited inflammatory macrophages to an anti or non-inflammatory state and potentially promote repair and limit chronic inflammation. Models of myocardial ischemia, as well as hind limb ischemia, which is a model of peripheral artery disease. And I will be using human correlates to look at how these subsets change and how they may predict outcomes within our heart failure patients. And so with that, I'm going to finish by thanking my incredibly supportive supervisor, Dr. Slava Eppelman, and the entire Eppelman lab, specifically my students that I had the privilege to work with that I've mentioned throughout, Hamira, Anthony, and Sarah for all her help on the bioinformatics work, a number of collaborators and mentors, as well as my funding. And with that, I'd be happy to take questions.
Thank you so much. That's just an absolutely amazing amount of work, you know, in terms of trying to decipher this paradox that we always had, right? In terms of the macrophages being protective and reparative, but at the same time, you know, can be actually inflammatory and damaging, right? You know, and how does one actually, you know, sort of reach that balance? And I think you begin to really, you know, help us to delineate, right, the origins of their and the changes through environment and through development. Yeah, so this is really exciting. Yeah, so
thank you.
So I don't know if folks have questions. So the way we're doing this is that if you have questions, you know, we would very much invite you to use the microphone here. Yes, okay. And but also please use the Q&A feature, and which then will actually come up on my phone here. Yeah. So maybe we can actually, yeah. So get started. Yeah. So Tom, go ahead.
Yeah, that was a great talk. Very fascinating. So when you get into the myocardial infarction model, and there's this sort of yin yang between the resident macrophages and the incoming tissue or source macrophages, and then there's an initial proliferation, but then in the adult myocardial infarction, there's a lowering. So there must be some cell death pathways happening. Is that intrinsic, maybe part of the proliferation that's linked with apoptosis or cell death pathways? Or do you think it's coming from these other infiltrating macrophages, maybe limiting the number through also activating cell death?
Yeah, so we do know a lot of cell death happens. The drastic loss of the resident population probably comes to cell death due to that oxygen deprivation. But we also know that this proliferative response happens even in the absence of that cell death. So in models of sort of hypertension or viral infection, for example, that stimulates the resident populations to begin proliferating. And that appears to be specific to the disease phenotype, whereas in the homeostasis, there really isn't a lot of proliferation happening, and macrophage numbers are held to like a very defined number. So I think it's probably a bit of both happening, responding to the injury and trying to withstand the death themselves. Thank you.
Sarah, that was excellent. Thank you for sharing all that data with us. So related to, so I have two questions. One is related actually in similar line to what Tom was asking. Have you looked not at the death so much of the macrophages, but their egress? So either to lymph nodes or elsewhere in terms of them just not being there, but them leaving?
We've looked a little bit and we don't see them leaving, but we certainly haven't extensively looked at that. But our initial thoughts are no.
Okay. And then the second question is, so, you know, obviously there's lots of data sets, I guess it's another two-part question, data sets related to single cell seq in especially murine hearts. So have you done sort of a meta-analysis or whatever you want to call it to you know, other conditions, looking at your signatures and these things in those whole, because a lot of, I think yours are mostly just the macrophages you've done single cell on, not the actual cardiomyocytes and fibroblasts and things?
Exactly, yeah. Yeah, so we've looked at that quite a lot actually. And the trick is we're trying to do some of our own analysis, but you have to do single nucleotide to get the cardiomyocytes in there.
Got it.
So it's a bit more of technically challenging in that sense, but we have gone and looked at some of the published stuff, and what we see in terms of the macrophage populations, it's pretty consistent, and there's a few papers out now that have done single cell RNA-seq at different time points post-MI, so we can kind of look at this progression a little bit, and we see a lot of our data being recapitulated, and then we also seeing some new populations popping up both early and later. So when we picked day 11, it was mainly because we were interested in catching a time after that initial inflammation. So we hadn't those residents, but sort of before all of the recruited were gone too, because we wanted, so it was kind of trying to find that middle ground. But now that we can sort of look at that massive recruitment and that initial inflammation, which is actually mostly dictated by neutrophils, and then we can see the populations come up. And interestingly, there's a few populations of recruited macrophages that start to take on a anti-inflammatory, maybe M2, although we don't use M1, M2. So it does sort of look like they kind of phenotypically change a little bit. The resident ones kind of stay the same.
So the reason I was kind of asking that question is about this potential influence on fibroblasts and endothelial cells. And are you starting to look at that, like either from computationally through other data sets, like how your, you know, interested macrophage populations could be either, you know, secreted factors or turning on programs that then influence the fibroblasts or the endothelial cells, those kind of things?
Yeah, so we're just starting to look at that. And I mean, we were mainly focusing, I guess, on fibroblasts at the moment. And we do see they're responding, but don't have too much on that yet.
Okay, thanks.
I can help to follow up on that. In fact, the fibroblast fate almost follow the same kind of, you know, so in terms of the, you know, sort of the resident, you know, fibroblast versus, you know, the transformed fibroblast. Yeah, so it's fascinating.
Yeah, and they kind of inform each other too, which is kind of interesting and intricate.
Yeah, exactly. So this is fantastic. So maybe I can ask, because there are questions, you know, coming up as well, and there are people raising hands. So obviously generate a lot of interest. So maybe this is actually from Lynn McGinney.
Awesome.
And here comes a tough question. Yeah, this is a fascinating talk. Do you have any idea in terms of how the macrophage population can actually repress the cardiomyocyte hypertrophy process? You know, so just a simple question.
Yeah. So I mean, it's kind of a difficult one, because in the setting of MI, I showed that loss of the macrophages increases hypertrophy, but actually in the setting of hypertension, the resident macrophages are critical actually for the hypertrophy to occur. And so we don't really understand why this happens and why there's this differential response, if it's adaptive versus maladaptive remodeling. So yeah, we're not really sure, but we do know that IGF-1 plays a role.
Go ahead. This is a really great talk.
Thanks.
I have two questions. The first is kind of on the flip side of what you've been looking at. Are there diseases that, you know, where you might see a proliferation or a massive expansion of the resident macrophages, and like if that can inform some of your results?
Good question. I don't know of any scenario where we have sort of I don't know if you're asking like a disease
or condition or
condition
yeah,
of this.
I can't think of
I mean,
either.
we see instances where the resident macrophages do proliferate and expand. So neonatal cardiac injury, for example, in hypertension, the resident macrophages do start proliferating and expanding. So we do see a response to injury expansion. But in a pathological sense, cancer maybe. Yeah.
And my second question is about one of your last sort of aims for your research program. So in your pseudo-time analysis, where you basically see the monocytes leading to the CCR2 macrophages and those CCR2 macrophages kind of linking to the other populations,
yeah.
I'm wondering if there's any like metabolic programs that you've seen that could be intervened in, or like environmental factors that you may be able to, to target to be able to shift sort of the fate of those, or at least, you know, not necessarily the fate, but what they resemble in their functional capacity.
Yeah, so we know the ISGs and like inflammatory macrophages tend to take on more of a glycolysis. So for sure, we haven't really looked at that, like mining our data for it. Other people are looking at that though, and it's a really great point. Yeah.
Maybe we could actually see if one of the hands up participants virtually could actually join us and ask the question directly. And this is Dr. Ferribos Soheli. Oh yeah, please make sure you unmute and then ask the question if you can. Yeah, go ahead. Oh, sorry. Microphone is actually active. Apologies. Yeah, so we'll work on that as a work in progress. And so we have a question for Morgan Fullerton. Yeah, so amazing work and congratulations. And do you have any sense as how I guess this is a question on metabolic pathways of these three overarching groups are programmed, and in terms of whether, you know, is there anything known in terms of the difference, you know, amongst them?
Yeah, again, we haven't really looked at that, but it's a really great question. I'd love to talk more and dig into that. Yeah.
Okay, so. Yeah, so
I see some in the chat.
Yeah, so there's, oh, okay, yeah, so maybe I'll ask this one first. So you're right, you know, so because they're coming through a fast and furious, different sources. So this one is from Mihae Wei, she's interested in your BAM brain macrophage subpopulation. Do you have any information on how they functionally differ from, say, the microglial cells?
Yeah, they seem to be quite different actually. And the BAM field is kind of new and exciting and quite diverse. So they, depending on where they reside, I guess is the answer. So it's very different from microglia. And they seem to be playing a role in the regulating that immune blood-brain barrier. And they're present in the meninges, they're present in the subventricular spaces. So depending on where they're located, they have very diverse functions actually. But I'm not an expert on that at all. But so yeah, it's a very diverse, very exciting field.
Yeah, so we'll talk about some of that. Yeah. Oh, yeah. And so maybe we'll, in view of time, just 2 more questions that I just found there in the chat, right? And so one is from Michelle, I think it's Arganino. Yeah. So the question is, so it's a very cool stuff. So do you know when in evolution did the macrophages start to acquire these common features? And are there any environmental cues that drives the differentiation?
Yeah, so as far as the first question, when in evolution did they acquire, I don't know the answer to that. We don't have a lot of data on macrophage subsets in sort of lower vertebrates, for example, but I do think there was a zebrafish paper out recently that was looking at the macrophages, so I'm sure we will come to find that. And in terms of the second question, common environmental cues, so we are thinking it has a lot to do with the subtisular space. So they really listen to their environment very well. And 2 things that pretty much every tissue has are blood vessels and nerves. So the prevailing theory is that one of the resident populations is sitting next to blood vessels, talking to the endothelial cells and acquiring their phenotype and playing a role in the endothelial cell homeostasis. And the other subset is sitting next to nerves and playing a role in innervation and that stuff. So it's a theory, but right now that's our favorite one.
That's favorite of ours as well. Yeah, so we're totally biased here. So yeah. Maybe the last question then from Hank Kim. Again, amazing work. And so and congratulations. Wondering at the transcript level, can you see tissue-specific macrophage signatures within the self-renewal and the resident macrophages? And do these signatures, exist embryonically?
Yeah. So if transcript levels, were they able to see? So yeah, so the tissue-specific macrophage signatures are absolutely there almost as soon as they enter the tissue. Is asking if they have a signature specific to self-renewal. Yeah, yes and no. Like all macrophages have the ability to proliferate. It is more the lack of replacement. And I think this has maybe less to do with the macrophages themselves and more to do with the access to the niche. So the niche hypothesis being that the monocytes will come in to fill an empty niche if an empty niche isn't available. But the easiest way to fill an empty niche is for a neighboring macrophage to proliferate and fill up that niche. So instead of expressing cytokines to recruit a monocyte in to fill that empty niche, they'll just initiate a proliferation response. And so it has more to do, I think, with the environment cues, the local availability of the growth factors such as CSF1. And then sort of there's this CSF1 threshold, I guess, that it needs to reach in order to increase or to enter the proliferative phase.
Yeah, okay.
Hope that answers that question.
Yeah, I think that is the essence of the question. Yeah.
Yeah.
So I think this is fantastic. So it's just in view of the time, we kind of have to wrap up this exciting discussion. But it really illustrates, I think, the amazing, I guess, sort of the discovery in terms of how the macrophages, really is multifunctional. And, so that not only is embryonically critical, but, it really actually determines, how the organ functions, right? Both in physiology and also in pathological states. And so I think it behooves us, to really actually understand how this interplay works and their kind of a social environment, and so that we can actually modulate in favor of a good outcome, for example, under injury. Yeah, so this is really exciting, I think, so look forward to, of course, more interactions with our team here for the rest of the day. And hopefully, you will be able to set up even more collaborations on forward going basis. Thank you so much.
Great, thank you, everyone.
And thanks for joining us virtually. Yeah.